Hello everyone, and welcome back to Oxen Academy. In today's video, we're going to be talking about the types of transport. And to do that, we have to take a look at this term right here, concentration gradient. Concentration gradient is essentially an analysis of the concentration of molecules or solute within a given area. So let's take an example and a look at this situation right here. We have this enclosed area right here, and we can see that on one side, on this left side right here, the concentration is high. Yet on the other side, concentration of these molecules is very low. And a key thing to define within the concentration gradient is essentially the flow with the concentration gradient and the flow against the concentration gradient. Flowing with the concentration gradient essentially means that this area with a high density amount of particles will over time move to this area with a low density and spread out evenly to create a sort of equilibrium in which everything is then dispersed evenly and throughout the entire area. Flowing against the concentration gradient, however, is where this particle and these particles on this side, on the right side, with a lesser amount in density, actually move toward the side with the higher number of particles. Something that's important when we define these next two terms of active transport and passive transport. The main difference, well, there are actually two main differences between active and passive transport, is that active transport uses energy, while passive transport doesn't use energy, so no energy. And at the same time, active transport flows against the concentration gradient. Flows against, meaning that the area with a lesser amount of particles and a lesser density moves toward the area with a higher density, while passive transport moves with the concentration gradient. In today's video, we're going to be going over the types of passive transport. And really, there are three main types. We have diffusion. We have facilitated diffusion. And lastly, we have osmosis. And I'll highlight passive transport so that everybody knows what we're talking about exactly here. So let's get right into it. Diffusion is the very bare bones idea of passive transport. Essentially means that, well, particles flow to the concentration gradient, with the concentration gradient. It's the movement of particles, from an area of high concentration to an area of low concentration. And let's just take an everyday example of this. Let's say that we spray Febreze within our house. So we spray it in this area right here, and we have a bunch of these Febreze scent molecules within the small area of our house. Over time, from this high concentration area, these Febreze molecules are going to spread out in every which way, moving to the areas of a low concentration, and in this case, no concentration, so that the Febreze molecules are dispersed very evenly. This is why you'll see if you spray any scent within your house, in a short while, it smells, well, really intense in a certain area, but after time, it disperses throughout your house, giving a more widespread yet less potent smell of this very same thing. That's the idea of diffusion. Now, let's look at facilitated diffusion. It essentially keeps the same idea of a movement of particles from high to low. Yet this time we have to go a bit back into where we talked about the cell membrane and transport proteins. If you take a good look at transport proteins, it looks something like this, with most cases a channel in between these two sides of a protein, and of course we have the lipid bilayer on either side. In this case, we talk about how we have these molecules, let's just say on the outside of the cell, in high concentration and low on the inside of the cell, these high concentration molecules actually flow to the other side of the cell in order to create an equilibrium. So essentially, it's the movement of particles from high to low, yet in this case, with a transport protein. So with a transport protein. That's the only real difference between diffusion and facilitated diffusion. So what's an example of facilitated diffusion? Well, one example is actually everybody's favorite glucose. Glucose. In many cases, it can be hard for glucose to pass through the cell membrane, especially given the fact that it's a pretty big molecule. It's actually a 6-carbon molecule that really can't fit through the lipid bilayer. So in this case, it has to take the help of a protein channel, a transport protein, to get to the other side. Because these protein channels are a bit bigger than this lipid bilayer and the spaces in between them to let this glucose get through. And lastly, we have osmosis. Now, osmosis, as defined, is essentially the diffusion of water, and very specifically water, through a semi-permeable membrane until the solution, or this area of water, until the solution reaches what we call a dynamic equilibrium. What exactly is a dynamic equilibrium? Well, let's think of it like a seesaw. We have this fulcrum right here, and we have the two separate sides. Yet in a seesaw, you can never be perfectly balanced. This can really never be a straight line and have it still like that 24/7. Yet when we're on a seesaw and we want to be balanced, we have to make slight changes here and there, which means that at sometimes this side's going to be a bit higher, and for that we have to compensate and drop a bit lower. But sometimes you get a bit too low and the other side gets a bit too high, and then they have to drop, something like that, in which we don't have a perfect equilibrium at all times, but we're always making slight changes to ensure that we are straight on with the other person. So let's draw this within a solution. We have a little beaker here. I'll draw this in blue. And we have some water right here, and we have this semi-permeable membrane. So we'll draw a couple of dashes to know that some things can get through, some things can't. Within this, we have a couple of solutes, or some molecules that can't pass through this membrane. So we have a couple, let's just say 4 solutes on this side and 1 solute on the other. So the problem with this is that there is a low concentration of water on this side in comparison with the right side. So what will happen is that water would actually go through the semi-permeable membrane to get onto the left side, which essentially creates a solution that looks more like this. Have a high concentration of water, low concentration of water on the right side, and it would fit perfectly with the number of solutes. Now, it might not seem like an equilibrium, but the concentration of the water is directly proportional to the concentration of solutes. And taking a look at all of these, if we go back to this idea of a passive transport, we can see that all of these really fit in with this idea of going with the concentration gradient. At the same time, it's important to note that none of these use energy, making all of them forms of passive transport. Thank you for coming to this video, and I hope that you all have a wonderful rest of your day.